Welcome back to Lucid Science. This week, we'll be looking at CRISPR-Cas, Biology, Mechanisms, and Relevance by Hill and Charpentier. First, let's go over some terms. A prokaryote is a microscopic single-celled organism that has neither a distinct nucleus with a membrane nor specialized organelles. Prokaryotes include bacteria and cyanobacteria. A eukaryote is any cell or organism that possesses a clearly defined nucleus. This includes animals, plants, fungi, and protists. Viruses are infective agents that insert their genetic material into their hosts and are only able to multiply within the living cells of this host. They are also known as phages. Homologous is being the same or equivalent. In genetic terms, this means containing the same sequence of genetic material. CRISPR-Cas9, where CRISPR stands for clustered regularly interspaced palindromic repeats, and Cas9 is its relative protein, is a prokaryotic adaptive immune system that memorizes previous infections by integrating short sequences of invading genomes, termed spacers, into the CRISPR locus. Okay, I think we're ready, so let's get into it. Prokaryotes have evolved several defense mechanisms to protect themselves from viruses. The ability of the CRISPR-Cas9 system to target DNA sequences using programmable RNA has opened new avenues in genome editing in a broad range of cells and organisms with high potential in therapeutic applications. This review summarizes major discoveries in the CRISPR-Cas field, discusses the role of CRISPR-Cas in prokaryotic immunity, and describes applications of the system as a DNA editing technology. Being the most abundant organisms on our planet, bacterial and archaeal viruses are a constant threat to prokaryotic life. The prokaryotic immune system, CRISPR-Cas, caught increasing attention in the scientific community, not only because of its unique adaptive nature, but also because of its therapeutic potential. The CRISPR-Cas system acts in a sequence-specific manner by recognizing and cleaving foreign DNA or RNA. The defense mechanism can be divided into three stages: adaptation, biogenesis, and interference. In adaptation, a distinct sequence of the invading macrophage, called a protospacer, is incorporated into the CRISPR array. This event allows the prokaryote to memorize the intruder's genetic material and displays the adaptive nature of this immune system. To enable immunity, the CRISPR array is transcribed or copied into a long precursor CRISPR RNA that is further processed into mature guide RNA that contain the memorized sequence of the invader. In the last stage of immunity, mature CRISPR RNAs are used as guides to specifically interfere with invading genetic material. This process stores the specific genetic sequence, but disables it so it cannot damage the prokaryote. Let's look at the significance and applications of this process. The use of CRISPR-Cas in therapeutic approaches has become increasingly relevant in different fields of medicine. Engineered CRISPR arrays have been designed to kill disease-causing bacteria by targeting and disabling genes that cause antibiotic resistance or severity of the disease. However, the medical potential of the CRISPR-Cas system goes beyond antimicrobial treatment. The introduction of efficient and precise modifications into the genes of an organism is the basis for genetic engineering. Because of its simplicity, effectiveness, and the possibility to target multiple genomic sites simultaneously, use of the CRISPR-Cas9 system is usually favored over other known systems. The bacterial defense protein Cas9 is used to target almost any desired DNA sequence with the help of a targeting RNA. Repurposing the CRISPR-Cas9 system for genomic editing uses the DNA repair mechanisms of eukaryotic cells. After the introduction of a double-strand DNA break, the cell can repair the damage by non-homologous end joining. However, this method is inaccurate. Precise genome engineering relies on the homology-directed repair pathway, where a piece of engineered DNA that shows the same sequence is used to repair the DNA and insert the desired sequence. This can be seen in Figure 2a, where Cas9 cuts the DNA sequence, triggering DNA repair, and leads to the insertion of the desired sequence. The applications of this new technology are astounding. Precise remodeling of the genome can be used to cure genetic diseases. Scientists were able to repair mutations that cause cystic fibrosis, as well as fight against HIV and cancer. Interest in the field of CRISPR-Cas has rapidly increased in recent years. Numerous studies shed light into the underlying processes of the adaptive prokaryotic immune system, revealing its potential in modern medicine. However, despite the potential that lies within CRISPR-Cas9 technology, further investigation is required to make it a safe tool for therapeutic use. And although some challenges remain, it seems to be only a matter of time until CRISPR-Cas9 based genome editing will be used in a variety of therapeutic approaches. And that's how CRISPR-Cas9 is leading the way in our fight against genetic diseases. Find us on social media at Conservation Made Simple, and I'll see you on the next Lucid Science.